Hello College Biology. This is our first video in our protein series, and I hope to cover the basics of proteins and their monomers. So, to start, proteins are made of the 5 of the 6 elements of life, hydrogen, carbon, oxygen, nitrogen, and sulfur. This is the first time we see sulfur, and that is because some of the monomers have sulfur, and some of them do not. But none of them has phosphorus. So there's that. And then proteins are amphipathic. So they have regions of polarity and regions of non-polarity. And so let's get to it. Let's talk about those monomers. This is a series of the monomers of proteins. These are called amino acids. And you can see they are a wide variety. When we had the monomers of carbohydrates, all those polysaccharides were made of glucose, so there wasn't any variation in the monomers. And then when we went to nucleic acids, there was a little bit of variation in the sugar, because if it was RNA, it was ribose, and if it was DNA, it was deoxyribose. But there was some variation in the nitrogenous bases, because there were 4 or 5 possible nitrogenous bases that you could have in your nucleotide sequences, and you would still be able to make your polymer. Well, these are even more varied. There are parts of the amino acid that are the same for all amino acids, but then there are parts that vary, and these different parts define the amino acid. You can see that they all have names: glycine, phenylalanine, tyrosine, arginine. And so these names are based off of this variable portion of the amino acid, which is called an R group. So let's take a look at, well, before we take a look at these parts further, you can see that there are 20 listed here. Now, there are actually many more amino acids. It's estimated that there's about 200 or so amino acids, but only 20 of them are used in protein polymers. And so, since this is a biology class and we are studying right now protein, we're only going to be concerned with these 20. So these are our 20 amino acids that are used as the monomers to make protein. So let's take a closer look at the parts that are the same. Every amino acid has a similar structure. It has an amino group on one side, which is NH3 positive. It's got a nitrogen bound to 3 hydrogen. We have the central carbon in the middle, which is sort of the hub that all of the other parts connect to. We have on the opposite side the carboxyl group, which is 1 carbon that is often shown as a double bond to oxygen and a single bond to an oxygen. So it's a carbon bound to 2 oxygens, and there's a negative charge over here. And then we have the R group. The R group is the variable portion. So all of this is the same for every amino acid, but the R group can be different for each amino acid, depending on which amino acid it is. So these R groups are divided into different properties. Some of them are non-polar. Some of them are made of polar covalent bonds, so they have partial charges, they are polar. And some of them have full charges, like an ion. So they have ionic charges here, this negative, negative, and positive, positive, and positive charge here. So they vary vastly in their polarity, and that matters. That is what allows proteins to be amphipathic. Some of the amino acids they are composed of are nonpolar. Other amino acids they are composed of are polar. And so some regions of the protein are polar and some regions are not, and so it is amphipathic. Let's go on. How do these amino acids get put together to make that polymer, to make that protein? Well, that's done by a ribosome. Ribosomes are what are responsible for linking amino acids together to actually construct a protein. So how does that work? Well, very basically, the amino group of one amino acid and the amino group of one amino acid and the carboxyl group of another amino acid undergo a condensation reaction, and you now have a peptide linkage. You have a peptide bond. Now, a peptide bond is a normal polar covalent bond, but it is always occurring between a carboxyl group and an amino group in order to form this particular link. And now you do many of these together, that gives you your protein polymer, which we will discuss more in detail in the next video. So let's review. Protein are composed of the elements of life: hydrogen, carbon, oxygen, nitrogen, and sulfur. They are amphipathic, and their monomer is the amino acid. Each amino acid is composed of 4 regions: the amino group, which is our positively charged NH3, our carboxyl group, which is our negatively charged COO, and our central carbon, which is the carbon in the middle that's connected to everything. Additionally, we have R groups connected to that carbon. The R groups vary and define the amino acid. There are many R groups, there are many amino acids, but only 20 are used in protein as protein monomers, and some of those amino acids are polar and some are nonpolar. To go from the monomer to the polymer, the ribosomes link the amino groups of one amino acid with the carboxyl group of another to form a peptide bond. We're going to go more into what is entailed in that and what happens to that polypeptide in our next video. I hope this helps. Struggle well.